when we started on this, I wasn't really thinking it would be a long-term thing necessarily. So we were saying, okay, well, we need a tool for this orientation determination part.
Right.
And in crystallography speak, that's called indexing.
That sounds like the story of so many open source projects, doesn't it? You start with creating something you need right now, then it becomes useful for others, and suddenly a lot of people are using it. Which is what happened to Tom White, my guest for this episode, who talks about the software tool he created and which is now used in material sciences and biology, a tool called CrystFEL. Before we go into the ins and outs of CrystFEL, I'd like to set the scene a bit. Because in order to appreciate what the software does and can do, it's important to talk about the research fields it's meant to be used in. CrystFEL helps to understand the structure of materials, and not just crystals, where part of the name comes from, but also biological matter, for instance proteins. Understanding the three-dimensional structure of materials is important because it gives us a clue how they behave in different environments and under different conditions. A good example is the molecule hemoglobin, which is in our blood and responsible for the transport of oxygen through our body. And the way it does that is through what is known as a conformational change, that is, changes in its structure when it binds or releases oxygen. And if there's a problem with the structure of the molecule, it can lead to some serious health problems like, for instance, the human sickle disease. In short, the physical arrangements of atoms, ions or molecules in materials play a key role. One way to study them is to hit them with a powerful light source, like X-rays for example. And this is a technique that has a very long tradition, which is down to the fact that X-rays, or indeed light in general, interacts with matter. X-rays, when they hit an atom, or rather the electrons in the atomic shell, change their direction after impact. This is called scattering. But because of the weird and wondrous nature of the subatomic world, the world described by quantum physics, X-rays act both like particles and waves. And waves, when they overlap, well, they can either add up to give bigger waves, or they can cancel each other out, which means when you capture the X-rays after they hit the material probe, you should see a spatial distribution of intensity peaks and spots of low or no intensity, which is called a diffraction pattern. And it is from that you can, in a way, reverse engineer, with the help of some clever maths, the three-dimensional structure of the material at hand. The double helix structure of the DNA was discovered this way, after all. But since the early days of X-ray diffraction, the way we analyze the experimental data has changed, of course. Data from X-ray diffractions are collected electronically these days, and in order to collect and analyze them, you need software, which is, of course, where Tom White's set of tools called CrystFEL comes in. I met Tom in his office at DESY end of last year, 2025. DESY is the German Electron Synchrotron located in the city of Hamburg and home to a very powerful source for this kind of research, as you will hear now from Tom.
So my name's Thomas White. I'm a staff scientist here in the Photon Science Scientific Computing Group at DESY. Before I moved here, I was at the Center for Free Electron Laser Science, also on the DESY campus.
Oh, right.
Yeah.
So you've been at DESY for a while now?
For a very long time, yes, since 2009.
Oh, right.
So straight after my PhD, I came here.
Where was the PhD?
PhD was in Cambridge, and it was in electron crystallography in the Department of Material Science and Metallurgy. So probably any other university, the same PhD would have been in the physics department, but in my
Cambridge
case, it was in the
wasn't.
material science department.
Yeah.
So
Well, fair enough.
Yeah, that's fine.
And you also play theatre, you said?
Yes, yeah. So
do
my
some
main
sound.
outside work, I'm doing a lot of theatre stuff, mostly lighting, sound.
Are you the
main
sound
thing.
engineer then?
Yeah, sound and light, yeah.
We can exchange some war stories maybe at some
Okay.
stage. But actually the reason we're meeting today is a program called CrystFEL, and FEL stands for femto electron laser.
Free electron laser.
Oh, free electron laser.
Yeah.
Right. So maybe we can start with that, because before we go into the nitty-gritty bits of the software, maybe you can tell us a little bit about the background, what it's used for.
So CrystFEL is a suite of programs for processing data from serial X-ray crystallography experiments. So the primary application of this is in structural biology, so looking at protein crystals. Myself would definitely not self-identify as a biochemist, for sure. But the technique is also used for other samples like material science things and also small molecule chemistry type samples. But the main application is definitely in structural biology at the moment. So serial crystallography is a technique for measuring diffraction from crystals, small crystals. It was originally developed at X-ray free electron lasers. So hence the name. Exactly, yeah. So hence the name CrystFEL. So the point here is that you only get one frame of data per crystal. So you have a stream of crystals being passed into the path of the X-ray beam, and the X-ray beam is basically continuously on or continuously flashing at whatever its repetition rate is. And whenever this X-ray beam hits a crystal, you get a diffraction pattern on a two-dimensional detector. So it's an image that comes out.
What comes to mind is the early study of DNA by Watson and Crick. And there was Rosalind Franklin, who was also deeply involved with that. And I think they used a similar technique to discern the structure of the helix, didn't they?
Exactly. There's a bit of historical perspective here, because serial crystallography is in contrast to rotation crystallography. And if you went and asked a structural biologist, how do you measure X-ray crystallography data, they would say they use rotation crystallography. And in this, what you're doing is you're taking one crystal and you're putting it in front of the X-ray beam, and then you're continuously measuring data while you rotate the crystal in a controlled way.
Right.
So you kind of sweep out a three-dimensional volume of data. Now, when you go to an X-ray laser, you only can get one exposure per crystal, because the X-ray beam is so powerful that it destroys the crystal within...
It grills it, basically.
It completely explodes it and it vaporizes it. It's gone. So you never have any chance to get more than one exposure per crystal.
Yeah.
At the free electron laser, you absolutely have to use this serial crystallography technique, because there isn't really any other way. But since then, it's kind of been developed towards synchrotrons, so a synchrotron radiation source. So particularly the very latest generation of synchrotron radiation sources are also very powerful. It's very intense X-rays. And then you start to need this kind of technique as well. But if you go back into history, before the rotation method was really established, people were actually acquiring data in this kind of snapshot, single frame mode.
All right, okay.
Yeah.
So we're kind of stepping back into the single frame mode.
Yeah, so I've heard one person in the field has commented that actually the period of time of history where we do rotation crystallography could actually just be a kind of a blip. That remains to be seen, I think.
Yeah. Here at DESY, you have quite powerful lasers.
Yeah, so we have kind of everything on the campus here. So, of course, there's the synchrotron radiation source Petra 3. So that is, I think, still the most intense third-generation X-ray synchrotron source in the world.
In terms of, could you give us some numbers?
So the way the intensity is measured is in terms of the number of X-rays, so the number of X-ray photons over a period of time in a certain spot size.
So luminosity.
And within a certain bandwidth, so a certain range of wavelengths. So you sort of combine, you divide by all those numbers, and you get this number. But Petra 3 is a third-generation synchrotron, and now we have fourth-generation synchrotrons. I should say synchrotron radiation source, really, because I don't know if the particle physicists would agree with calling that, because it's a storage ray.
And you also have a different laser.
Yes. So there's FLASH, which is a free electron laser. Now, FLASH is right here outside the building where we're talking to you now.
I climbed over that in order to get to this building.
So that produces these very short X-ray pulses, very intense radiation. But the wavelength of FLASH is long, so it's kind of more in the sort of ultraviolet region of the spectrum. If you want to do crystallography, you need real X-rays, so like 1 angstrom wavelength. And for that, you would need the European European XFEL. And that starts here on the campus, so you might have walked past it. The experimental hall for that is over in Schenefeld, so about 3 kilometers away.
Yeah.
And the underground tunnel for that crosses the state line out of Hamburg into Schleswig-Holstein.
I didn't know that, actually.
It's well worth a visit.
It's quite an experimental setup. I was working not very far where the campus of the University of Hamburg is, so back in the days. So the experiments, are you still involved in the experiments, or are you looking exclusively after CrystFEL?
I'm mostly just doing the software these days. When I started this, so when I was at CrystFEL, I was doing a lot of experiments. So there was a period of time when I would travel to at the time, the only operating hard X-ray free electron laser in the world, which was the LINAC coherent light source, which is in California.
Yeah.
So there's a time when I was going there multiple times per year. So I kind of decided that I wanted to focus more on the software and rather than just concentrating on my own experiments, actually build something that can then be used for many people, many experiments.
You said you're material scientist officially through Cambridge, but
by
actually a physicist.
Undergraduate degree, yeah. So I'm not so interested in the samples, like the biology. What are we actually looking at? What kind of enzymes? What kind of reactions? I mean, it's not that I don't think it's important. It's just that it's not my personal interest. So what I'm able to do is just concentrate on the software, making sure that the analysis methods, the algorithms work as well as possible, that the software is as easy to use as possible, because biochemists don't have that much time to learn how to use all this stuff. I still have contact with the experiments, of course, because people are sending me emails, asking me questions all the time, often asking me to take a look at their data to figure out a problem. So I kind of keep my finger on the pulse of what's going on.
What made you actually go into the more software side of things?
I think it's kind of always been actually what I was destined for. I've always been a
It was
person.
written on the walls.
I programmed computers since I was old enough to basically sit at a keyboard. I think it was always the way it was going to go. I also just enjoy studying the methods, what's going on in the numerical methods or the physical processes. I just find this interesting. Yeah.
Did you start CrystFEL then?
Yes.
So what made you start it? Right now we're coming to CrystFEL finally.
Yes. Straight after my PhD, I came to DESY. That was just as the LINAC coherent light source was starting up. So the very first experiments there, this was like December 2009. And my group there was involved with these experiments. So we were thinking about what kind of data was going to come out of this experiment and how we would need to process it. The first thing I did actually was to start doing some simulations. So I created a folder for the source code for my simulations, and I just thought of a name of it. I thought, crystallography, FEL, CrystFEL, that'll do. And the name kind of stuck. Yeah, so the very first code for CrystFEL was written within a couple of weeks of me coming here, and it's just built up from then.
And that was initially simulation.
Yeah, the first thing was simulation. Since then, it's definitely expanded to analysis and processing of the data, and eventually most of the simulation code has actually been taken out now. That's because it didn't really compose very well with the rest of the code. Because simulation is kind of a different task to processing.
What exactly is the simulation? What do you simulate? The detector?
Yeah. So we have a model of the detector that's going to receive the X-ray. So the position, how large is it? What are the size of the pixels and so on? And how far away is it from the point where the crystals and the X-ray beam are going to intersect? So that's our origin. And the simulation is then to say, with the crystal in this certain orientation and the beam coming from this direction, what will we see on the detector? So where will the Bragg peaks form? So the diffraction of X-rays from a crystal means you get sharp peaks of intensity that are called Bragg peaks. And we need to know where those are on the detector and how intense they are.
Okay, that brings us to the detector, which we haven't talked about. What kind of a detector is it?
For detecting X-rays, it's mostly basically a piece of silicon. And then when the X-rays go through the silicon, they liberate the electrons from the holes in the silicon, and then you pull the electrons with a strong electric field into a detector, and then you detect the blip of current.
Okay.
Yeah. It's that times 1024 by 1024 pixels or larger. Yeah, there are different ways you can do it. At the time we started the experiments, actually, if you think about how you detect the X-rays from a free electron laser, it has to be a detector that works like a bucket. Because all of the X-rays are going to come within a few femtoseconds. So you kind of have a bucket that you fill up with these X-rays, and then you read out the strength of the signal. So that's what we call an integrating detector. If you're working at a synchrotron radiation source, the X-ray photons arrive with actually a little bit of time between them. I mean, it's a very, very small amount of time, but you can actually measure this, and you can actually count the photons as they come in. So that's what we call a counting detector. You certainly can't use a counting type detector at a free electron laser because it'll just see one peak.
And that's it.
Yeah, all the X-rays will arrive in one blast. So actually making a detector that can do this integrating behavior and can also keep up with the speed of repetition of the X-ray pulses coming from the free electron laser, that's actually quite an electronic challenge.
Were you involved in that as well?
Kind of tangentially. So the detectors that we're using are often research projects in their own right.
Indeed.
Yeah. So there are a photon science detector group within DESY. This job is to develop this kind of thing. That means a lot of the data we get that basically we have to deal with, we're using CrystFEL, is not necessarily the cleanest. So there might be defective regions of the detector. There might be variations in gain across the detector, or variations in gain over time between pulses. So that's all the stuff we have to deal with. And I guess that's another reason for kind of not needing the simulations as much, because it's so important that we look at what's really happening in the data.
But still, you need to have some kind of simulation in order to test things out. And how did you approach to actually modeling a detector of that kind?
I think the response part is fairly simple. So like how many counts of intensity do you get in your data array that you then read out into your program per photon. That part's relatively simple to model. What is a complete pain for us is the geometry. A lot of these detectors, to make them work fast enough, what they do is to split them into small panels, so little sub-detectors. There's a detector, for example, operating at European XFEL at the moment that has 128 separate panels. So it's a two-dimensional plane, just then split up into 128 little tiny submodules. And they're all reading out in parallel. So there's multiple network cables going into the back of this machine. And to process the data, we need to know quite accurately where these modules are in space, the coordinates within this plane, but also where they are in 3 dimensions. So if there's a tilt relative to the beam, we have to know about that as well. And that's been a very big topic for us recently. So this is probably sounding familiar from particle physicists that you've looked into. Yeah, so they also have detectors consisting of much more than 128. There we're talking, thousands. So what we've been doing recently is actually using an algorithm and, in fact, a piece of software borrowed from high-energy physics for this refinement task. So given our approximate model of the detector and where the panels are, we then acquire some serial crystallography data, and during our processing we can say, okay, well, we expected to see a Bragg peak in this location on the detector, but we actually saw it displaced by maybe half a pixel, a few pixels. So if that happens a lot, we kind of think, well, this panel should have been moved a little bit.
Which brings me to the next question, because I do remember we used Monte Carlo simulations for our detector, for instance, for estimating systematic errors, etc. But we had a library available from which you could build certain components. But did you start from scratch, or did you actually have something in place where you can model like the geometry of the detector?
We did have to start from scratch, unfortunately.
Yeah.
And it's still a problem to this day. So these complicated multi-panel detectors first emerged at the X-ray laser sources. They've since kind of expanded to synchrotron radiation sources as well. This means that the software for processing the data, not just crystallography data, but all kinds of data, if you're using such a multi-panel detector, you need software that can deal with this. And there isn't any, basically. So CrystFEL was one of the first to do this, and there was an experiment that's seared in the minds of many of my colleagues from about, I think it was 2012, the first time we met the detector with 64 separate panels, and we were sort of all going mad, just trying to figure this out.
So a program had to be written one way or the other.
Yeah. And now other experiments are facing the same problem. We've been collaborating recently with another beamline that does it's still crystallography in a sense. It still involves crystals, but it's not the kind of crystallography that I've described. They now have a multi-panel detector, and the problem is their software just can't deal with it. So we've actually provided a workaround for them based on CrystFEL to enable them to handle their data.
But detectors is not the only thing that CrystFEL is doing.
Yeah, not at all. Yeah.
Exactly. There's the data collection side, and I guess maybe also the analysis side.
Yeah, so CrystFEL will take you from these images. So we're acquiring many tens of thousands, hundreds of thousands of these individual exposures.
Yeah.
And each one of them is showing the diffraction peaks from a separate crystal in a separate random orientation, because there's no correlation between the orientations of the crystals. And for each one of those crystals, what we then have to do is determine, so first of all, where are the Bragg peaks? So this is like an image analysis kind of algorithm. And then what is the orientation of the crystal?
Right,
so,
which is a little bit trickier because you don't really know that.
Yeah, so there's been quite a lot of algorithms for working this out. They all basically come down to finding 3 dominant periodicities, so repetition in 3 dimensions. So even though you only have a kind of a 2-dimensional like shot exposure from the crystal, there's kind of difficult to explain without drawing a diagram, but it relates to a curved sphere of data. So if you know the wavelength, you know what this curvature is, and then turns out you can still find these repetitions in 3 dimensions. It's even easier if you know something about the crystal. So if you know the symmetry of the crystal, so symmetry is extremely important in crystallography, one of my favorite subjects. Or if you know what are the repeat distances for the crystal lattice. So if you know those, you're left with only 3 parameters you have to find, which are the 3 rotation angles of the crystal.
So it helps you with the data collection from the detector. It helps you with the analysis of the data, like, for instance, the orientation of reconstruction of the crystal in a wide sense.
So once you found the orientation, the next part is to then calculate, okay, where should we have seen peaks in the diffraction pattern? Now, the reason for that is that the thing that we ultimately want to measure is the intensities of these Bragg peaks. And the intensity can be 0. And if it is 0, we want to know that. We're not just interested in the peaks that we see, we're interested also in knowing at this point in the diffraction pattern, we should have seen a peak, but we didn't. Therefore, it's telling us that peak is actually weak, low or 0 intensity. So we calculate where we should have seen these spots, and then we measure the intensities at all of those locations. And then we're still not done, because across the tens of thousands of patterns, what we're then going to do is merge the intensities of all of these separate measurements together and make one final 3-dimensional data set. So because we knew the orientation of each crystal, because we worked it out, we can then form this 3-dimensional volume. So if you remember at the beginning, I mentioned the rotation experiment where you rotate the crystal to kind of sweep out this 3-dimensional volume. We're ending at the same point, but we're doing it by random orientations, putting these random orientations into the 3-dimensional box together.
Indirect. You're simulating the rotation in a way.
Yeah, shoot first, ask questions later. It's one of the ways it's been put. Yeah. So once you've got this 3-dimensional volume, there's some tools in the suite to calculate some figures of merit for that. So how good is your reconstruction? And then the next step is outside of the scope of CrystFEL then. So then now there's other software for this part, which is to then determine the structure of the protein or small molecule chemical or whatever it was you were looking at.
You've been working on CrystFEL for a while, obviously, but how did it all evolve? What's the history of that?
Programs were added as they were needed. So they started with a simulation that was later removed because of the availability of real data. There's a main program for this process that I just described of determining the orientation and then measuring the intensities. So that's called indeximagig. The reason for the kind of funny name is, well, when we started on this, I wasn't really thinking it would be a long-term thing necessarily. So we were saying, okay, well, we need a tool for this orientation determination part. Right. And in crystallography speak, that's called indexing, because it gives you these things called Miller indices, which are the 3-dimensional coordinates for the spots in the And I said, okay, well, we're going to need some kind of indeximagig.
And hence the name.
Exactly. Yeah. For merging, there's a tool called process_HKL. But as the methods kind of evolved, we learned better ways for doing this merging, this combining of the data, right? Because it turns out you can do more than just taking an average of the measurements. You can actually do some modeling. So the simplest part of this is, well, let's say you find one pattern where all of the Bragg peaks are a bit stronger than you would expect. So this could be because that X-ray pulse was a little bit stronger, a little bit brighter. It could also be because the crystal was bigger, or it was more well ordered. So the better ordered the crystal is, the stronger the diffraction will be. Now, when we're combining our data together, we want to kind of compensate for that. We want to say this pattern was a bit stronger, this one was a bit weaker. We scale the strong ones down a little bit, we scale the weak ones up a little bit, and then we merge them together. So that's the simplest kind of modeling you can do, but there's much more you can do that accounts for like the diffraction physics that's going on inside the crystal, as it's interacting with the X-ray beam. So that led to a new program for merging, which is called partialator. As it stands, there are actually 2 merging programs in the suite. And then there's various other tools, like this one called Compare HKL, this one called Check HKL. So these are figures of merit tools. The geometry refinement thing, that's a recent tool called Align Detector. We're kind of back to conventional names. And then on top of all of this, there's a graphical user interface. Right. And this is a relatively recent addition. People had been talking about it for a long time, saying,
Yeah.
oh, you should write a graphical user interface because, like I said, people concentrating on structural biology, they have a lot to think about. How to make their samples, how to clone the DNA for the whatever protein.
Did they do that by filling out a text file with input data of what they wanted to have, or how did that work beforehand?
For the user interface, it was, it still is really a command line based software. So all of these different tools with these names, the names I just told you are actually the things you type on the command line. So Unix based command line. The GUI, the graphical user interface, that was kind of a pandemic project. I gave a workshop right before the first pandemic lockdowns in Sweden. And it was, in a way, an epiphany because I was talking to these people who were using my software and they were showing me all the things they have to think about. They have to know about NMR, nuclear magnetic resonance spectroscopy. They're doing chemistry. They're doing whatever, wet lab work, all of this stuff. And then on top of that, I come along and say, okay, now you have to learn how to use my software. You also have to learn crystallography theory in order to understand all the symmetry considerations. And you also have to learn how to use a command line. And you also have to use a high-performance computing batch system.
And by that, they lost the will to live.
Exactly, yeah. It's too much. I kind of decided at that point, okay, I should just write a GUI. Let's just do it. It's worth spending time on. And then, of course, came the pandemic lockdown. So, perfect.
There was time for that.
There was time, yeah.
Let's talk a little bit about the software itself. Is it written in a particular language, or are you using different ones? I guess with the GUI, there comes a point where you have to use different.
So it's written almost entirely in C, including the GUI. Not for any particularly good reason, other than that it's what I knew when I started.
Even the GUI?
Even the GUI, yeah.
Do you use some libraries?
Yeah, it's using GTK, again, because I knew it at the time. In addition to the C code, there's also some scripts to help you, like maybe convert file formats or plot graphs of your data in certain whatever ways you might want to do. And a lot of those are written in Python. And just recently I added bindings to Julia. CrystFEL itself consists of a shared library core. So it's called libCrystFEL, and that contains all the routines for doing the things I talked about. So for like indexing a diffraction pattern, or for finding the peaks in a diffraction pattern, or loading an image with a multi-panel geometry, this kind of thing. And then the command line programs are built on top of that, so they use this library to do their work. And what I've done is to allow use of that library from Julia.
Why?
It makes it easier to put the parts together in a different way, like maybe a way that I hadn't thought of. Maybe someone wants to do something else. It's quite common that someone emails me and they say, I have this particular analysis problem. I've acquired data in maybe a certain new way that they've devised to measure the data. And they say, to analyze this, I need to process it in this way. I need to correlate this with this or whatever. And it's just something that's not in CrystFEL directly. And they say, could I add a feature to do that? And I say, well, I don't have time. Unless a lot of people ask it, of course, then I will do it. But by adding the Julia bindings, well, then people can actually put these pieces together and do these kind of things.
Which brings me to the question about on what kind of computers CrystFEL is run. Can it run actually on an ordinary computer, on a laptop, on a Macintosh, on Windows, on Linux?
Not on Windows. But
not Windows, but Mac and Linux?
It's supported on Unix-like platforms. So Linux is the primary development platform, so which is my computer here is Linux, Fedora. Most people are using CrystFEL on a high-performance computing system of some kind.
Right.
So,
and I thought that was the reason for Julia, the programming language.
I guess. I mean, I just like the language with Julia.
Okay.
And people have been asking for Python bindings like for a long time, and there have been some attempts. I'm not really I can write Python. I can certainly read it, but I certainly don't have the skills to do Python bindings for the library. It's actually really hard to write bindings from a C project to a...
I know that some have bindings from C to Python and that it's possible, but
it's
I'm not
certainly
sure
possible.
how much work is actually involved in
It's
that.
difficult because there are different memory management models. So the way you allocate and free objects. So for example, in CrystFEL, there's a structure that represents an image, right? A frame of data. And a frame of data can then contain crystals. So this is a data record of a diffraction pattern in this image. And there can be multiple of these. There can be, usually there's only one crystal, one diffraction pattern per frame, but there can also be multiple ones overlapping. And then that crystal then contains a list of Bragg reflection intensities. So that's another structure. And that's a list structure which then contains reflection items. So this all works well in C, because I can just write in my API documentation, here's how this works, here's how to free it, here's what's owned by what.
Yeah.
But Python and Julia are garbage collected languages.
Yes.
So that means there's a different memory management model where something goes out of scope, it will immediately be freed. And lining those two up really requires intimate knowledge of how things are working on both sides.
Was it that easy for this kind of mapping to Julia?
No, it definitely wasn't easy. Yeah. But it seems to work. Famous last words.
Yeah.
It works well enough. So like I say, most people are using it on the high performance computing system.
Right.
And this will be the system at the facility where the data was collected. So all the X-ray laser facilities and most synchrotron radiation sources have some sort of attached computing system.
DESY certainly has.
Yes. It's quite rare these days to download your whole data to your home institution and then process it there. Yeah, it's too big. It's too unwieldy to download it. Some people do, but I think that's kind of going out of fashion. So they're processing it on these systems, and then they're downloading this final result, this three-dimensional merged data set.
Right.
And then they'll work on models of the protein or whatever they're looking at. They'll probably do that part at home.
Is that what is called the CZP4?
Exactly, yes. Yeah, so CZP4 is one of the packages for solving protein structures from diffraction data, amongst many other things. I mean, we don't have any kind of analytics or telemetry on this, so we don't know for sure. But on the basis of what I hear from my ear to the ground, I think most applications of CrystFEL are on these high-performance systems. But I think most installations, are actually on Mac.
Right,
And this is because people will maybe take a sample of their data and they'll download it to their computer to work on, on the plane or whatever.
Right, yeah.
And that will be a Mac.
Yeah, which has Unix-like commands.
Yeah, it's a Unix system. So yeah, we are supporting CrystFEL on Mac.
What amazes me is it sounds like a very complicated program and quite involved, and it certainly keeps you busy. How many other people does it keep busy? Are there any other people involved in the software development and maintenance?
It's mostly a one-person project.
I.e. you.
I.e. me, yeah. So I've been tremendously fortunate because very soon after I arrived at DESY, I was promoted to a permanent staff scientist position.
Right.
So that's given me the freedom over the years to just work on this and not have this pressure to have to get the next big, whatever high impact paper or whatever.
Yes.
I can just focus on the software, and that's in fact what my managers want me to do. And it's at the moment very well aligned with what DESY wants to do in the upper management. So I think that's very comfortable. Apart from me, so there are contributions. So, for example, one of my colleagues is working on it's a web-based electronic metadata logging device. So it records the data the information about your data runs while you're measuring them. So what was the wavelength? What was the sample?
Okay, yeah.
What time did you start and end? Are there any notes, like something you noticed during the experiment? And this has a database of all the runs. And this program is calling CrystFEL to actually trigger the data processing.
Oh, right, okay.
That person is then sending me patches when he finds something that's not quite working correctly.
So is CrystFEL then an open source project?
Yes, yeah, it's GPL 3 since the beginning.
The question then is, it's open source, so people can download it and people could work with it, but the contributions, how do you manage that? Is that something that you review before you say, okay, I'm going to put that into core CrystFEL?
Exactly. There are sporadic contributions from all over the world over the years that CrystFEL has been public. So the first publication was 2012, the first actual release. So that was when it stopped being an internal lab thing and started being something that anyone can download. That was a long time ago. So the number of people and institutions that have contributed in some way, I think it's over 20 now. I wouldn't say I'm being rushed off my feet with contributions. I would quite like to be, but I think people they're concentrating on their own research. They don't necessarily have time.
And I guess it's probably also quite a beast by now to get into. I mean, how easy is it to get into?
So it's about 100,000 lines of C, if I remember correctly, from the last time I ran the line count on it. The people who work on it, they've given me positive feedback about the quality of the code. So although it's written in plain C, so not C++, it is written in quite an object-oriented way. So we have encapsulation as much as possible, separation of concerns. I try to follow all of the rules of software engineering. We do have continuous integration, so we have tests, we have unit tests. We're building on multiple different systems on every commit just to check, and that includes Mac. I think as a software project, it's okay. It's just some of the stuff you have to understand to work on crystallographic code is just quite hard.
You have to know something about the field.
Yeah, there's a lot of theory that's very important. I tend to be quite pedantic.
In what sense?
If someone's adding a feature, that feature should work for example, all the possible crystal symmetries. And that requires considering why it might fail for some symmetry class. And that needs to be done, to completion. And that sometimes is asking quite a lot from someone who's just added a little feature for their PhD or whatever.
So you have to turn people down sometimes.
Sometimes. I think it's less about turning stuff down, more about just recognizing when something belongs as a private contribution.
Make a fork of that and add it.
Yeah. Let's say a new way of acquiring data, and that requires some specialist analysis method. That should only be added to the core software if it's going to be used by lots of people.
Yeah,
right?
yeah.
So first of all, we kind of establish that before rushing to add everything. It's also about the support, right? The long-term support.
So that was my question, actually.
Yeah. So what I found to be quite a good strategy, actually, is to have external contributions be incorporated by means of a shared library themselves. So, for example, last year, I think it was, maybe the year before, a group in Switzerland came up with a new indexing algorithm that actually uses GPU. And they wanted this to be part of CrystFEL, and I agreed because that sounds good. Fast indexing is what we want. That was built as a shared library, and then CrystFEL can depend on that shared library. And then the maintenance, it's clear that the maintenance of that shared library belongs elsewhere.
Right. It's almost like a plugin.
It is a plugin, yeah. It has another advantage, which is that it can be reused in other software. There are, I don't like the word competitors, but there is other software for serial crystallography, and they could also use this indexing algorithm because it was a shared library. So I think that's a positive way to go.
So I'd like to wrap up with what's in store for CrystFEL, and also in the view of how can you sustain the work on it with more and more people getting interested in it?
So, like I say, the support by means of my permanent position has been fantastic. I think if it wasn't for that, the software would probably never have been released in the first place because my postdoc would have finished.
That would have been that.
Yeah. So that didn't happen, which is, I can only be grateful. Sure, right? I haven't been that successful in getting extra funding. So applying for grants. I have been applying for grants. It's been hard. I think it's hard for everyone, but I think it's particularly hard for software first kind of projects to get funding this way. The Helmholtz Association, which is the overarching organization that supports DESY and other research centers, actually has quite good support for software, at least recently.
They have a competition, I think, or
the Helmholtz Software Award, Yeah, so CrystFEL was nominated for that twice.
Oh, right. Okay.
The results of the last call are still not out. But there are opportunities within Helmholtz to get funding, so I've been applying for some of those.
Funding in the sense of getting someone else to help.
Yeah, so this will be for another full-time person to work on the code. In the future, so at the moment, CrystFEL is concentrating on X-rays, but one thing that people are doing recently is to acquire serial diffraction data, but using electrons. So this is in an electron microscope, transmission electron microscope, and they have very similar looking data. And to process that within CrystFEL is actually already possible. There are some special considerations, though. One is that the optics of the electron microscope have distortions. So the diffraction pattern that you see will kind of be twisted in certain ways, will be wonky. Yeah. And those need to be kind of accounted for in the same way that we account for this multi-panel detector geometry. So I think it's a very similar problem. I think it's amenable to this method from high-energy physics. So that's kind of the next thing that I'm very interested in doing. And also just generally improving the support for electron diffraction. So that means things like the file formats that they use, support for the box that asks for the X-ray energy, the photon energy. So this is something like 9,000 electron volts per photon. That's what the number that can be then converted into a wavelength. But you can't do that for electrons. Or you can, but it's a different calculation. You say I accelerated the electrons to 300,000 electron volts. What is their wavelength? Then it's a different calculation. This stuff has to be put in the software somehow. Yeah, that's some of that's already done, and some of it's still to be done. People are getting results with electrons, which I'm very happy about.
Well, we'll wait with bated breath for the outcome from the Helmholtz competition.
Yes.
And in the meantime, I thank you very much for your time, Tom.
Yeah, thanks for coming.
And all the best. If you're interested, and I hope you are, you'll find the links to CrystFEL and some background info on X-ray diffraction in the show notes, as per usual. Next week, we look into the workings of a very different kind of material, our brain, because I had the pleasure of working on a very exciting project during my time at the University College London, building an online brain atlas. The primary investigator, Eugenio Iglesias, worked on this project for a long time, and recently the study has been accepted for publication. And together with my ex-colleague James Hughes and Eugenio, we dive into this project once more. I hope you'll tune in then. But in the meantime, thanks for listening. Oh, time's up. See you next time. But before I forget, this podcast is covered by the Creative Commons license. See ya.